PFAS webinar, ORDA Science Webinar 101 series, organized by KCB Snyder and recorded by her via Microsoft Teams on November 14th, 2024 at 19:02 UTC.
And again, this guide was put together by USGS folks and US Fish and Wild folks, to Fish and Wildlife, not wild folks, but they could be, but wildlife service, to basically introduce people that may not have PFAS background to some key concepts for when you're about to go sampling, the analysis, and so forth. So I will go ahead and start. I'm going to turn this off for right now, and then we'll move forward.
PFAS and the Quest for Clarity, Navigating the Sampling Saga. Official logo for USGS, Science for a Changing World. Report title, Guide to Pure and Polyfluoroalkyl Substances, PFAS. Sampling within Natural Resource Damage Assessment and Restoration. There is a QR code that leads to this guide, also called the Open File Report 2024-1001. Scan the screen with your phone or select the link in the transcript for access. Official logo of ORDA, U.S. Department of the Interior. Erin L. Pulster, USGS. Sarah R. Bowman, USFWS. Landon Keel, USGS. Jeffrey Stevens, USGS. United States Department of the Interior, United States Geological Survey. Most images referred to in this presentation are linked to their citations in the transcript.
Okay, so again, I'm going to go through basically the document. If you haven't seen the document itself, it's broken up in a number of chapters, but I'm going to introduce you briefly to some of the nomenclature and physiochemical properties of PFAS, their production, use, and status right now, the sources that they've been used in, fate and transport, and introduce some of the key concepts when we're talking about atmospheric deposition, soil and sediment, and biota. And then some of the considerations that should be taken into account when designing studies and moving forward in terms of baseline considerations, how to respond to spills in the environment, and ways to remediate, or things to consider when you're thinking about remediation efforts. And also the sampling design considerations. A lot of these overlap quite nicely, but I will point out and highlight some important design considerations, analytical methodology, and then what do you do when you get the data, and what does it mean?
Diagrams of two polyfluoroalkyl compound structures, PFOA and FTOH, as seen on page 2 of the sampling guide. These both follow the structure Erin describes for PFAS.
So PFAS, they're a very complex class of man-made compounds that are structurally diverse, consisting of about 16,000 chemicals that either have explicit structures or do not. There's a lot of definitions to PFAS, but one of the main ones that is consistent is they all have basically a functional head group and a fluorinated tail. They can be, the head group is typically hydrophobic and the tail, hydrophilic, and the tail is typically hydrophobic. They can vary in terms of carbon length from carbons that can have basically 1 to 14 carbons.
Table from Gorzonic Engineering showing that the root letters in the name of a PFAS can indicate the number of carbons it contains, the root based on the carbon backbone. For instance, the root but or letter B appears in PFBS and PFBA, indicating that they each contain 4 carbons. The root HEP or letters HP appear in PFHPA, indicating that it contains 7 carbons. The root Desc or letter D appears in PFDA, indicating that it contains 10 carbons.
And just to explain that a little bit further, some folks don't know that, that in terms of the carbon length, a lot of people don't know that hidden inside the name of the PFAS, you can kind of decipher how many carbons are in there. So for instance, PFBA is a butanic acid and it has 4 carbon lengths, whereas PFHPA has 7 carbons. So this is sort of a little trick if you don't know how many, how long the carbon tail is. You can kind of look at the name and decipher how many carbons are in there. They're also broken down by whether or not they're short chain or long chain. And of course, those are even different by class. So there's the PFCAs, or the carboxylic acids, and the sulfonic acids, and those differ by the number of short chains. They just have it broken down a little bit differently. But that goes into, so in terms of the family tree of PFAS, it's considerably long and complex. And this is just an abbreviated family tree that separates the 2 main classes of PFAS, which is the non-polymers and the polymers.
Image of the PFAS family tree, adapted from ITRC 2023.
Most of us are looking at PFAS in the non-polymer section, or analyzing for targeted PFAS. And those are the most common ones, again, are the carboxylic acids, the PFCAs, and the sulfonic acids, PFSAs, which are your common PFOA and your PFOS. Through NGenX, that's another pretty common one that people are looking for. This is one of the short chain, 6 carbon length alternative compounds. Some of the main physiochemical properties of PFAS, again, is they contain both the hydrophobic head group and the I keep mixing that up, hydrophilic head group and the hydrophobic tail. These properties basically make them, they also have some compounds are basically either one or the other, hydrophobic or hydrophilic. PFAS are both. And so what that means is they're going to partition differently throughout the environment. One of the main differences between PFAS and some of your legacy compounds like PCBs or PAHs is PFAS binds to proteins as opposed to lipids. So when you're looking for PFAS, you want to make sure you're looking for those protein-rich tissues if you're looking for them in biota. Some of the main properties that exist in PFAS chemicals is that they are very durable and resistant to stains, oil, and water. And because of these properties, they also are very resistant to biodegradation, photooxidation, direct photolysis, and hydrolysis. So that's kind of where they got the term forever chemicals. So some of the common uses and sources you might be aware of, they're pretty much everywhere. They're in our nonstick pots and pans. They're in pesticides and herbicides. One of the big sources is the AFFF foams, the fire foams. They're in our food packaging. They're in personal care products, paints. You name it, they're probably in it. They're in our clothing and our textiles. They're used pretty much everywhere. So the timeline, basically, they were first synthesized back in the 1930s, late 1930s. And then by the 1950s, they were starting to be manufactured and commercially produced for things like Scotchgard. You might have heard Teflon was one of the first ones. They were starting to be used in food packaging in about the 60s, and our nonstick pots and pans came out thereafter. In the early 2000s, we started phasing out PFAS and PFOA. It's important to note that these phase outs, at least in the US, were voluntary. By the phase out, there's only about 8 companies or so in the US that voluntarily phased them out. So even though they're phased out, they still might have some reservoirs somewhere and some within the states, and there may be some companies still manufacturing or using them. China still uses them, so we are still exposed. In terms of environmental consequences and health consequences, the effects in both fish and rodents of PFAS were known since about the 1950s. Human blood, it was detected in human blood as early as the 1970s. And then our analytical improvements in terms of analysis of PFAS, they started getting better and better in the 2000s. And then in the late 2000s, the 2010s, and onward, we started developing more regulations. Where are we with the regulations? 2024 was kind of a busy year. The EPA and the GSA are ensuring that now custodial contractors are required to use PFAS-free cleaning products. These are all in the Federal Registry, by the way, if you want to look up the documents. The EPA also released interim guidance on how to the destruction and disposal of PFAS that is not in consumer products, so more building products. And more importantly, they issued the first national primary drinking water regulations up here. So this essentially shows the maximum contaminant level for PFOA and PFOS is 0. That's the goal. That means it's not enforceable, but they would like to see 0 PFAS and PFOA concentrations in our drinking water, but the maximum contaminant level that's enforceable is 4 parts per trillion for both of those individually. They also released guidance for 3 more, PFNA, PFHXS, and GenX, and those are each 10 parts per trillion for goal and the maximum contaminant level. There's also a mixture where you can look up, combine these, add 2 or more of these together to determine whether or not they're above the hazard index of 1 or below. They also released the freshwater aquatic life criteria for PFAS and PFOA. These are nearly 6 orders of magnitude higher than our drinking water concentration. So here we're looking at about the acute water concentration and water column for PFOA is about 3.1 ppm. And also the aquatic life benchmarks for 8 additional PFAS have also been released. These are also in the ppm range. And there's also now acute saltwater life benchmarks, aquatic life benchmarks, and these are also in the ppm range.
PFAS fate and transport diagram, as seen on page 9 of the sampling guide.
So fate and transport of PFAS in the environment. Essentially, the consensus is, since there's so many products and uses of these things, the main primary source or primary producer is industrial facilities. So those companies that make them, 3M, DuPont, those sorts of things. And then there's what we call primary sources, which consists of the consumer goods, the firefighting chemicals, landfills, and wastewater treatment plants. It's not that they're making these, but these are basically a primary source of PFAS, where they end up essentially and what is releasing them to the environment. And then we have the secondary sources, so soil, groundwater, secondary sources, meaning they're entered our soil and our sediments, and now they can enter into our receptors, such as wildlife and humans. So as I mentioned, PFAS is pretty much everywhere. We're finding it in our surface water. We find it in deep water. We find it in fish, soil. Pretty much every environmental media has had PFAS detected globally.
Diagram, overview of PFAS sources and environmental pathways, De Silva et al. 2021.
So atmospheric deposition. Atmospheric deposition is a pretty important topic in terms of PFAS. The atmosphere is considered a sort of reservoir or long-term accumulation of PFAS, but it also is responsible for the short and long range transport of PFAS around the globe. So essentially, once it's emitted from industry or wastewater or landfills, however it gets into the atmosphere, you might have a volatile precursor that goes through processes such as oxidation that turns it into a terminal product or another mediator that ends into a terminal product like PFAS or PFOA. But anyway, they can go through the atmosphere, they can transfer long distances, they are deposited through wet and dry deposition, so rain, snow, dust. It can enter in, again, all throughout the globe. Once it's in the surface water, it can also come up through aerosols and sea spray and then continue the cycle.
4 global heat map type images. Spatial modeling of PFOA indicate. Thackeray et al., 2020.
Oops, this was a duplicate, but here is a image of some modeling that Thackeray et al. Had done of PFOA in the atmosphere. And basically this graph here, A, is PFOA deposition across the globe. And what you'll notice is some of the highest concentrations are along the mid-latitude. These are PFAS greater than, with chain lengths greater than PFOA, so greater than 8 carbon chain lengths. And again, you can see sort of a pattern here. Basically, the deposition rates of PFOA and any PFAS that is being transported is going to vary depending on that precursor terminal end product formation rates and the precipitation rates throughout the globe. These highest deposition rates, again, typically occur over source regions or downwind of strong emission sources. So, and then what you see from a lot of the high deposition is in the equatorial zone, and this is primarily due to the high rates of precipitation and photochemical transformation throughout the environment. And this is just a graph of the differences between the short and long chain. It just kind of also provides indication about how long sometimes the transformation process takes. And so also the short and chain, this is a ratio of the short and long chains, but this also demonstrates how they're removed a little bit quicker with precipitation.
Diagram, fate and transport of PFAS in the Vadose zone. Sharifan et al., 2021.
If we get into soil and sediment and some of the characteristics that control PFAS transport in soil and sediment, it has to do with some of the soil characteristics as well as the PFAS characteristics. So for instance, the amount of carbon content in your sediments, the functional groups and chain lengths of your PFAS can really dictate where they're going to move throughout the subsurface. Some of these other aspects that integrate or determine where it's going to end up is these air-water surface interfaces, the interfaces between the solid and the water, so sediment particles and water. It can also be the non-aqueous liquid water interface. That is basically water immiscible organic compounds or like solvents or petroleum compounds. But basically PFAS in pore water is relatively low, but we typically see high concentrations of PFAS in that air-water interface or within these interstitial parts of the particulates. The long chain PFAS compounds tend to bind to the suspended particles, and therefore that's why a lot of times you'll see them in the sediments as opposed to the water column. And they're typically retained in more of the shallow surface soils, whereas the short chains are more mobile than the long chains, so they can tend to move down through into our groundwater.
Diagrams. Expected orientation and accumulation of PFAS at air-water interface, ITRC, and the environmental behavior of PFAS in aquatic ecosystem, Liu et al., 2019.
In terms of surface water, these also PFAS and their physiochemical properties also indicate where they're going to partition within the water column. Again, I mentioned there's both the hydrophobic and hydrophilic parts of the PFAS, so they're going to orient themselves between the air and the water. So in surface water, we see some of our high concentrations of PFAS at that microsurface layer, as well as in sea surface foams, because they basically, because of their dual properties, they create a surfactant behavior. So they tend to kind of stay up on that top of that water column. There has been studies where they're finding super high concentrations in the sea spray aerosols, and that can lead to more atmospheric transport throughout the environment. In the water column again, it's going to, where they partition is going to depend on basically the PFAS compound, the chain length, and the amount of suspended particles and organic content you have within the water column, because the long chains may bind to those sediment particles and fall out, similar to some other organic compounds that we see in the environment.
Diagrams. Removal of PFAS by wetlands, Arslan and Eldon, 2021. Bioacclimation of PFAS, Lewis et al., 2022. And bioaccumulative PFAS and precursors in fish, Pickard et al., 2022.
In biota, it gets a little bit trickier in terms of, it's most of it, for most biota, the exposure pathways are contaminated water, diet, and food. Again, these are proteinophilic compounds, so they mostly bind to protein-rich tissues, blood. Typically, the highest concentrations in organisms are highest in the brain and gonads, followed by liver, muscle, and blood actually is in there. So depending on the species, these might alternate. So that's why they're not necessarily in order of highest level. That's going to be species specific. Bioaccumulation within the environment also depends on chain length and species. So we do see some bioaccumulation of PFOS. We typically don't see a lot of PFOA in organisms, but we do see a lot of PFOS in the higher trophic level positions. Also, plants, for instance, you're going to see different partitioning between the roots and the above ground leafy portions. The long chain PFAS typically get trapped down here in the roots, whereas in the above ground leafy portions, you typically find the short chain PFAS. Also with fish, there's a lot of new data coming out on fish and the bioaccumulation potential of the long chain PFCAs, the PFSAs. So you can kind of see they increase until they reach about the carbon chain length of 10 or 11, and then they start to decrease. A lot of the associated effects with PFAS in wildlife and in humans, they share a lot of the health effects that include a lot of cancers, tumors. There's a lot of immunological effects, developmental and reproductive effects, endocrine disruptors, hepatic systems, neurological and cardiovascular effects as well. So the list goes on. In terms of baseline response and response and remediation considerations, when you're designing a study to, if there's a spill near a source or not near a source, one thing to take into consideration is, do you know what the anthropogenic background is? Basically, because these are synthetic compounds, there should not be any natural background levels. So understanding what that anthropogenic background is, because again, even though there may not be a source close by, we have to consider that atmospheric deposition that can add to this. Also characterizing the sources and environmental media. This is going to help also figure out, once you get all your data back, determine what the source is, or if you can, understand what the effects are, the levels, etc. This all takes into account, like, there's so many things that needs to be considered, like any organic compound, but understanding the degradation process and the precursors and the pathways. So if you get data back that has precursors, what does that mean? What are the spatiotemporal effects? What sampling techniques are you going to use? And what's the weight of evidence? Some of the response considerations, if there is a spill, how are you going to fight the problem? Are you going to fight it with the problem? Meaning, will you use AFFF? AFFF, although most of the formulations are being switched to alternative mechanisms, some places still have AFFF or may need to use AFFF if life is on the line. So if you do need to use AFFF, what should you consider? And typically that's mostly how are you going to contain your AFFF so you can clean it up later, and how are you going to deter the wildlife to reduce injury. And some of the remediation considerations is, for instance, there's some mines that they are trying to remediate, and they added biosolids to remove metals. Well, now we have another problem because now we have metals and we have PFAS. So just considering the different mechanisms to remediate an area and taking into consideration the PFAS content of biosolids and sewage sludge that typically is used sometimes to help remediate.
Diagram: PFAS accumulating in surface water foam from the Ecology Center.
So sampling design considerations when you're going out to sample. Again, I mentioned the common uses of PFAS. So when you're designing your study and getting your gear together, you have to take a close look of what your gear is made out of. Because, for instance, a lot of the water pumps or that people go out to sample water have Teflon liners and tubing and things of that nature, and those may contain PFAS. So understanding what is in your field gear is going to be critical for when you're out collecting for a PFAS contaminated site. Same thing with collection containers. Typically glass containers were used a lot for tissues or sediments, but there's some indication in the literature that some PFAS compounds actually bind to the glass. And then the recommendation typically is to use polypropylene. Polypropylene for large volumes of water is probably better for the field, so you don't have broken glass anyway. But there's also some indication that polypropylene or some types of plastic may actually leach PFAS. And then there's some PFAS compounds that actually bind to the polypropylene as well as the glass. So, but there's different ways to get around that when you get into the lab. The other things to consider when sampling is the weather and the seasons. So weather meaning, again, precipitation can be a source of PFAS. So do you go before or after a rain event or a snow event? Or wind, I bring up wind because if you're actually sampling in a lake, for instance, and the wind is blowing and you notice a pile of foam on one end, foam again, because we're here at the air-water surface, it accumulates high levels of PFAS. So you may want to actually be sampling the foam as opposed to over here. You might get a low bias if you sample here. So those are some sorts of things. Also, if you're sampling fish, depending on the question, you may want to sample after a rain event to get the highest recording level, or do you want to do a before and after? So some of these considerations has to do with your question in general, but to keep in mind that the weather will impact your PFAS concentrations. Location of sampling is basically where do we want to sample? That also has to do with which environmental media or matrix do you want to collect? That again, all has to do with your sampling question. Do you want to look at the partitioning of something within a particular ecosystem or environment? In which case you would probably need to collect everything from the surface water microlayer down to the sediment.
Venn type diagram of total fluorine, TF, Scheller et al., 2024. At the center is targeted PFAS. In a circle around that one is total oxidizable precursors, TOPs. In a circle around that one is extractable organic fluorine, EOF. In a circle overlapping with that one is adsorbable organic fluorine, AOF. In a circle around all of the circles previously mentioned is total organic fluorine, TOF. In a circle around all of these circles is inorganic fluorine, IF. At the top of this circle is the label total fluorine, TF.
Analytical methodologies. So this is advancing rather rapidly. Most people are looking at the targeted PFAS. Let me back up. There are different extraction methods for the different matrices: drinking water, non-potable water, solids, biological matrices, plant tissues, etc. EPA 1633 is for the non-potable water. EPA 533 and 537 are typically your drinking water. EPA 1633 is trying to include all of these, the non-potable, the solids, and the biological matrices. The differences between those, for EPA 1633, the extraction methods are pretty similar, and with the 533. The differences between those methods are basically the list of compounds and the detection limits. There's different analytical methods for evaluating whether or not you want or measuring their targeted PFAS. So those are a known list of PFAS. The EPA 1633 consists of about, I think, 40 or 50 compounds. Then there's also the TAPA, or top assay total oxidizable precursor. This is going to give you those PFAS precursors. Then there's the extractable organic fluorine, adsorbable organic fluorine, and total organofluorine and organic fluorine. So this would give you the full scope of what type of, or the extent of PFAS contamination you might have in an environment. But knowing what to do with all this and how to interpret it can be tricky. There are lots of contract labs, including, the EPA is also working on validated methods for pretty much all of these. I don't know about the inorganic fluorine, but most of these the EPA is working on validated methods for. USGS has methods for the targeted, the non-targeted, and I believe the EOF. The non-targeted is basically the ones that it's sort of like suspect screening, where we're looking for these fluorinated compounds but don't have standards to accurately quantify them. So determining which of these methods you want, you have to go back to what is your actual question, and will you know what to do with all this data once you get it?
Diagrams, PFAS, and Household Products, Diwapriya et al., 2023. Illustration of precursor transformation resulting in the formation of PFAS, L. Trozzolo et al. Comparison of PFAS sources, Langberg et al., 2022. Scatterplot of LPFOS, PFUNDA, and PFTRDA, Stoltz et al., 2023.
So now that you have this PFAS data, what do you do with it? So there's still a lot of research being done on how to fingerprint PFAS, but we're getting better and better and more clarity in terms of the different types of PFAS that are used in the different types of consumer products. We can also use a variety of multivariate tools to basically separate and determine between a consumer product and, say, AFFF, or consumer packaging, food packaging. There's different ways that you can look at that through multivariate tools. There's also some demonstrated diagnostic ratios similar to other organic compounds that might use say the long chain versus the short chain compounds. So there are some demonstrated diagnostic ratios. Most of the fingerprinting types of research still has a lot of uncertainty around it, just because we're still determining more and more PFAS in the environment through those additional analytical tools. You can use those additional analytical tools, or the multiple methods that I was indicating, like the target, non-target, and total, to sort of get a whole picture of what you might be encountering. Those different, if you have information on precursors and the terminal products, you can sort of look at recent deposition versus old deposition. So recent would have a lot of those precursors, where as an old weathered source, let's say, might have more of the terminal end products. You can look at the distance between a source and where you're measuring to determine the same thing. Do we have a lot of precursors, or do we have a lot of terminal end products? Knowing what those pathways are, and there's a lot of literature coming out on the pathways, but that's going to be important to understand, is those precursor to terminal end products, which that's going to help you determine the source if that is your ultimate goal. You can also compare the profiles that you see in your fish tissues or your sediment to profiles that might be available for particular sources. So all of these things, basically you got to hit the ground, dig into the literature. There's also differences between the manufacturing process. So some manufacturing processes, you won't have the branched isomers of, let's say, PFAS or PFHXS. So that could also indicate what type of source you're dealing with, if you understand the differences between the manufacturing processes and the branched isomers or the isomers themselves. So with that, it's just sort of a brief introduction to the guide.
Acknowledgements. ORDA, USGS, Environmental Health Program, PFAS Integrated Science Team. 3 QR codes, scan the screen with your phone or select the links in the transcript for access. Guide to PFAS Sampling, PFAS Research at UGSG-CERC, and USGS PFAS Integrated Science Team. ePulster at usgs.gov.
Ended a little shorter than anticipated, but I wanted to acknowledge ORDA and USGS Environmental Health Program for funding. These are some important links. If you don't have access to the PFAS sampling guide, I encourage you to download it. It has the links to all the literature I mentioned, as well as EPA websites, ITRC that has a live website with updates on PFAS information. We have an extensive research program here at the Columbia Environmental Research Center in Missouri, and you can look up some of the research we do here on PFAS, as well as the USGS PFAS Integrated Science Team. We have information about our nationwide studies on that site as well. And with that, I will take any questions.
So far, nothing in the chat. Does anybody have anything they want to ask Erin while she's on the line?
Which slide? Sherla.
Maybe just the previous slide?
Oh,
Yeah.
Links to the pubs. Yep. Maybe. Here we go.
Well, I'll just ask a quick question, Erin. What do you think are the limitations of the current sampling techniques for PFAS?
Limitations. Some of the limitations are going to include consistency across all studies. I think for the most part, most people are pretty consistent in terms of using the polypropylene. I see there's a comment in there that you can rinse glass containers with silicone-based compounds. But in terms of sampling, I think the only limitation is making sure that you're pretty thorough in terms of what gear and collection tubes and things that you're using are adequate for the study. Make sure it's consistent with others across the basically standardized sampling techniques is what's pretty important.
Great, thanks for that. And it looks like we have a question from David. Has U.S. Fish and Wildlife Service, or USGS, brought any natural resource damage claims for PFOS family of chemicals yet?
Not that I'm aware of. I know there's been some statewide lawsuits, but USGS, since we're not really regulators or policy, we would not bring any claims. We do some research resource damage assessment, but we're not on the regulatory end of the scope. So that I don't know. U.S. Fish and Wildlife, I'm not sure if they have brought any cases, but I know there has been some state suits made, but I don't know of any others.
Thanks for that. I think Amy, our solicitor, chimed in that no, not at this time.
Oh, okay, good.
Next question is from Amy. If there's an event and a release, what would be your decision criteria for when and what to sample?
So that I think you'd have to first start with site characterization, GIS, like know what the area is and where the sources may be coming from. If there are no sources, then I guess where it spilt, you would want to, if it was soil, collect soil, sediment, vegetation, depending on how long that is, again, the short chain are going to transport more quickly down through the vadose into the groundwater, basically. So with that being said, knowing when the spill was and what to collect, if it's water, you're going to want to, I would suggest collecting surface water, water column, mid-water column, sediment. But again, because they're going to be partitioning because of the PFAS. Oh, I just noticed I spelled USGS wrong. Sorry, USGS. Sorry. But they're going to partition differently. So depending on the question, and if you know what the source is, what the compounds were used, then you can kind of figure out where to sample. But it gets tricky because you have to sort of know the source of the compounds and where it was spilled. So sorry, I don't have a real clean, direct answer, but it's really going to be site specific and problem specific.
Okay, next question is from Herb. I have read speculation that the incoming administration plans to roll back some of the recent PFAS limit rules implemented by the EPA. Have you heard anything about that or are able to speak on that at all, Erin?
I have not heard that, so I can't really comment on that. Sorry.
Okay. And then we have another question. Thank you for this presentation. Found it very useful. Would it be possible to get a copy of the slide deck? We are recording this presentation, so that will be made available to everyone. Erin, I'm not sure if you're planning on distributing the slide deck or if you want to just leave it at the recording.
I'm happy to do either. It's fine. I can also provide the document that the slide deck is based off of as well, if that would be helpful. Yeah, either way, I can do that.
Okay, great. Well, we'll make sure that's available to everybody that attended today then.
Looks like Suzanne Dunn has her hand up. Did you? Was that one?
Go for it.
Yeah, thanks. I was wondering if you go back to the slide on the mentioned using, when biosolids or soil amendments or sludge. I'm assuming the consideration would be to test those before use as opposed to not using them for, purposes of increasing carbon in soil on projects. Is that a fair statement?
Yeah, absolutely. Because different biosolids, sludges, different areas, they're all going to have different mixes of PFAS, etc. in them. So absolutely, I would recommend testing them. Some states, actually Maine, I believe, just banned the use of biosolids in the state of Maine because they were so highly contaminated. They are a considerable source. So yes, if that's the only option, I would definitely consider testing first.
Okay. And just as a follow-on, the comment you made earlier that they're ubiquitous in the environment. So, adding them to a site that already has a certain level of PFAS, I mean, is it consideration of like, we might not be adding to them because they're already there? So is it the consideration of just testing them and then making sure you're not creating an additional problem above what's already maybe out there, because again, kind of everywhere already? I don't know if that makes sense, but
yeah, I mean, I think what you're asking is like, if it's, should we use them if it helps the problem, sort of? Is that sort of where you're going? Or
yeah, I mean, if the goal of using these soil amendments is to increase carbon in the soil that adheres other contaminant issues that we're trying to address, and to build soil for restoration projects. However, that some of these products, soil amendment, sewage sludge or biosolids specifically, would have PFOAs in them. But what I'm saying is that we know that there's levels already out there in the environment, as you stated. They're ubiquitous, but they're not the same everywhere. They're right. So knowing what's currently on a site and then knowing what you might be adding to in the form of a biosolid to making sure that you're not adding to the site, but not to say that there's not some already there.
right, yeah. I guess once you add it, that's then going to be one, it's going to merge and transport to the groundwater, and it's also going to be taken up by crops and vegetation. So like, I know it's a big problem using biosolids right now on farmlands because it is being taken up in our foods. So I guess it all depends on, how that's a very good question. I guess evaluating the levels that are in your biosolids and taking into consideration like the compounds, which compounds are they that's in there, and is it worth adding, to utilize for a different purpose, but knowing what's in them. Sure, I think you would definitely want to know what you are adding for sure. Yep.
Thanks.
We have a question from Jean. You mentioned sampling guidance to avoid cross-contamination. Beyond that, is there a threshold of contamination above which safety is a concern for the person who is sampling? Is there safety guidance for PFAS and sampler safety?
Interesting. Not that I know of, in terms of the safety guidance for sampler safety. There's certainly a lot of guidance in terms of how to sample, what to wear to sample, what to do and not to do, what sunscreens to wear. Most of that has to do with like common sense, like don't apply cosmetics that have PFAS or sunscreen that has PFAS and then put your hand in the sample type thing. But in terms of safety guidance, I would probably refer to OSHA probably has levels in terms, I don't know if they must have, ATSDR might have levels on inhalation reference doses and things of that nature that might, if it is a source spill where you're expecting super high concentrations, I would probably consult ASTDR or OSHA to see what sort of inhalation levels they have there. Other than that, I have not seen any other safety guidance for samplers. Other questions? Show of hands of how many people are actually actively involved in PFAS sampling? There. See any? Anybody?
Well, great. Well, thank you so much, Erin. We appreciate your time. And we will take this recording and make it 508 compliant and then make sure it's available to everyone, as well as the materials that we discussed sharing. So thank you so much, Erin. And was there anything else? Yep. Thank you. Everyone have a great day.
Thank you.